Hello everyone, today I'm going to be talking about topic 7A respiration from the Edexcel IAL Biology Unit 5. So let us begin. What is ATP? The full form of ATP is adenosine triphosphate, which is known as the energy package used by the cells. ATP is used in muscle contraction, cell division, active transport, nerve impulses, etc. But more specifically, the energy stored in the third phosphate bond in an ATP molecule is released when ATP is hydrolyzed to carry out these functions. The ATP is synthesized in mainly two ways. First of which is substrate level phosphorylation, and the second one is oxidative phosphorylation. Substrate level phosphorylation occurs during the glycolysis stage. Now, what is glycolysis? Glycolysis occurs in the cytoplasm, where a glucose molecule, which is a 6-carbon molecule, by the way, the molecular formula of glucose is C6H12O6. So this molecule is phosphorylated twice using an ATP so that the glucose molecule becomes more reactive as it increases the activation energy so that more intermediate glucose intermediate is formed and the reaction starts. So this also ensures that the glucose molecules is not able to pass back through the cell membrane and out of the cell. In this process, the ATP is broken down to form an ADP and API. The 6-carbon compound after phosphorylation is now called 1,6-fructose bisphosphate. The fructose bisphosphate is split into two triose sugar or triose phosphate molecule, and this occurs by the action of the enzymes. The 3C triose phosphate is dehydrogenated, which means the triose phosphate now loses an hydrogen atom. So this hydrogen atom is collected by an enzyme called NAD, and as the triose phosphate loses a hydrogen, it is oxidized, and as NAD gains an hydrogen, it is reduced. So it is called reduced NAD or NADH. So after hydrogenation, the triose sugar is converted into pyruvic acid or pyruvate. A small amount of ATP is also made during the conversion of triose sugar into pyruvate. This number of ATP produced is actually 4 ATP. But only 2 ATP is our net gain, as we had used the 2 ATP in the beginning of the reaction, or in the beginning of the glycolysis stage with the glucose. So our net gain is 2 ATP. At the same time, as this glycolysis cycle is occurring twice, hence 2 reduced NAD is also formed in the process. So what is anaerobic respiration? Anaerobic respiration occurs when there is an absence or there is a very limited supply of oxygen. And anaerobic respiration takes place in the cytoplasm. During anaerobic respiration, the pyruvate that is produced in the glycolysis stage is converted into lactate or lactic acid by the enzyme lactic dehydrogenase. Now, to review a little of IAS course from Unit 1, enzymes are biological catalysts which speed up the rate of reaction without the cell being used up in the process. Enzymes have active sites which bind to substrates uh, that have very specific shape, and they have collision. Their molecules collide between each other and form enzyme substrate complexes. Moving on, in this process, NADH, which is the reduced NAD, is oxidized again back to NAD, which means that the NADH loses a hydrogen atom and thus becomes oxidized to NAD+. This allows glycolysis to continue and generate ATP even in the absence of oxygen. So lactate is a 3-carbon compound that can dissociate into lactate ions and hydrogen ions. This accumulation of lactate and H plus contributes to muscle fatigue and a drop in pH within muscle cells, i.e. the pH becomes more acidic by decreasing. So when oxygen is restored, lactate can either be transported to the liver to be converted back to glucose via the pericycle, or it can be oxidized back into pyruvate in the muscles, where it enters the Krebs cycle for aerobic ATP production. If there is enough oxygen present in the cell, the pyruvate molecule moves to the matrix of the mitochondria by the inner membrane of the mitochondria for the link reaction for aerobic respiration. If there is not enough oxygen present in the cell, the pyruvate molecule remains in the cytoplasm for anaerobic respiration. Also, the Krebs cycle occurs in the matrix of the mitochondria. In the matrix, there are lots of enzyme present that is suitable for this kind of reactions. What is the role of the link reaction and the Krebs cycle? The link reaction occurs in the matrix of the mitochondria, where there are enzymes present in the inner foldings of the mitochondria. The pyruvate that is produced in the glycolysis stage now moves into the matrix from the membrane of the mitochondria. The pyruvate inside the matrix is decarboxylated, which means it loses a carbon atom and an oxygen atom as well as dehydrogenated, i.e. it also loses a hydrogen atom. So the loss of carbon and oxygen forms CO2 and a 2C compound that is known as acetyl. And also the hydrogen atom that is lost is taken in by NAD, becoming reduced and forming reduced NAD, while the acetyl compound is becoming oxidized. Now acetyl joins together with an enzyme that is called the coenzyme A to form an acetyl coenzyme A. The energy contained in acetyl coenzyme A is released in Krebs cycle, and the carbon dioxide is removed by the decarboxylase enzyme from the side at the end of the link reaction. Also notice that during this stage, the link reaction, no ATP is formed. So in the Krebs cycle, acetyl-CoA from the link reaction combines with a 4-carbon compound oxaloacetate to make a 6-carbon compound called citrate. Citrate then follows a series of reactions like decarboxylation and dehydrogenation to return back to the original 4-carbon compound oxaloacetate. Firstly, the citrate is dehydrogenated to make reduced NAD by losing a hydrogen atom to NAD, forming a reduced NAD, and is also decarboxylated, which forms one molecule of carbon dioxide and thus forms a 5-carbon compound. Then the 5-carbon compound is dehydrogenated again to make reduced NAD and decarboxylated to release carbon dioxide again to form the 4-carbon compound. The 4-carbon compound undergoes further reaction. First of all, it goes through substrate level phosphorylation, which occurs producing an ATP or a GTP depending on the cell type. The 4-carbon compound is dehydrogenated again, but this time FAD accepts the hydrogen to form reduced FAD or reduced FADH. 2. Finally, the 4C compound is dehydrogenated one more time to regenerate oxaloacetate, forming the third reduced NAD or NADH in that process. Thus, through these reactions, 3 molecules of reduced NAD, 2 molecules of ATP, and 1 molecule of a reduced FAD are formed per cycle. These reduced coenzymes are then used in the electron transport chain to produce ATP. Also, remember that this step is occurring twice. So, as a result of that, in total, considering the two reactions, 3 molecules of reduced NAD in one step, 3 molecules of reduced NAD in another step gives us 6 molecules of reduced NAD and 4 molecules of ATP as well as 2 molecules of reduced FAD or FADH2. Now, let's move on to the next question. How ATP is synthesized by oxidative phosphorylation. Now, 2 reduced NAD from the glycolysis, 2 reduced NAD from ring reaction, 6 reduced NAD and 2 reduced FAD is formed from the Krebs cycle. These are the enzymes that are used in the electron transport chain as reducing agents. The electron transport chain occurs in the inner mitochondrial membrane, where there are proteins channel present that pumps the H plus ions in the mitochondrial uh, intermembranous space to create an electrochemical gradient. The electron transport chain begins with the NADH donating an hydrogen atom to the complex 1 of the ETC, in turn getting oxidized. The hydrogen atom gets split into H plus and an electron. The electrons then are carried from one complex to the other by the electron transport chain and mobile carriers. The energy released from the electron transfer is used to pump protons in the intermembranous space to create an electrochemical gradient across the crystal. The protons diffuse back by facilitated diffusion into the matrix due to the higher concentration and lower concentration outside the intermembranous space by the ATP synthase channel. Therefore, this releases energy that the ATP synthase uses to produce ATP from ADP and PI, phosphate ion. We are going to look now at the more detailed explanation of the electron transport chain by also observing the diagram. Continuing on the detailed steps of the electron transport chain. Look at the diagram here. So first step, NADH donating electrons to complex 1. In the first step, NADH donates electrons to complex 1. As the electrons are transferred through complex 1, the energy is released. This energy is used to pump H plus ions from the mitochondrial matrix into the intermembranous space and thus creating a proton gradient. After donating electrons, NADH is oxidized to NAD plus, which returns to participate in other reactions, while H plus ions remain in the matrix. FADH2 donating electrons to complex 2. FADH2 donates electrons to complex 2, unlike complex 1. Complex 2 does not pump protons, and electrons from FADH2 enter the electron transport chain at a lower energy level than those from NADH. So fewer protons are pumped overall. 3. Electron flow to coenzyme Q ubiquinone. Electrons from both complex 1 and complex 2 are passed to coenzyme Q, which is a mobile electron carrier in the membrane. Coenzyme Q shuttles the electrons to complex 3. Complex 3 and cytochrome C. Electrons are transferred from coenzyme Q to complex 3. As electrons pass through complex 3, more energy is released and additional H plus ions are pumped from the matrix into the intermembrane space. Electrons are then passed to cytochrome C, which is another mobile electron carrier. Complex 4 and oxygen as the final electron acceptor. Complex 4 receives electrons from cytochrome C. As electrons flow through complex 4, more H plus ions are pumped into the intermembranous space. At the end of the chain, electrons combine with oxygen, the final electron acceptor, and H plus protons to form water. This step is really essential because without oxygen, the ETC would completely stop, which would halt the ATP production. Now, moving on to the chemiosmosis theory, proton gradient and ATP synthesis. The proton gradient is created by pumping H plus ions into the intermembranous space. First, which is essential for ATP production. So, protons will naturally want to flow back into the mitochondrial matrix due to the high concentration in the intermembranous space and low concentration in the mitochondrial matrix. They flow through ATP synthesis, which is a protein channel, and this flow of protons is the chemiosmosis that provides energy to convert ATP and organic phosphate ion to back ATP. So, what happens to lactate after a period of anaerobic respiration in animals? Post exercise or running, due to the rapid need of energy, there is a rapid accumulation of lactate or lactic acid. This anaerobic respiration produces lactate or lactic acid from pyruvate due to limited supply of oxygen. This lactate accumulation contributes to oxygen debt, meaning after exercise, the body requires extra oxygen to metabolize the lactate. This restores ATP and replenishes glycogen stores. If sufficient oxygen is available, then lactate is converted back to pyruvate in the muscle cell, which then moves for the Krebs cycle for aerobic respiration, thus repaying the oxygen debt back as well. So, what is meant by the term respiratory question and the reaction of aerobic respiration as splitting of the respiratory substrate? A respiratory substrate is a substance that is used as a fuel and is oxidized during cellular respiration, i.e., aerobic or anaerobic respiration. During different respiratory substrates are used at different times in the cells of our body. Lipids are an excellent source of energy because they can be hydrolyzed rapidly to give fatty acid and glycerol. The glycerol is phosphorylated and thus enters the glycolytic pathway. Proteins are usually not used as a respiratory substrate. It is only broken down. Glucose and lipid stores are very low in our body, uh, as well as to say that when we are starving, that time the amino group in the amino acid of the polypeptides of protein must be deaminated to be used further for cellular respiration. Respiratory question is the relationship between the amount of carbon dioxide produced and the amount of oxygen used when different respiratory substrate is used in the cellular respiration. The amount of oxygen and carbon dioxide change depending on the activity of the organism, the type of food being respired, and other factors. We can calculate the RQ using the formula given below. If the RQ of an organism is greater than 1, then the organism is respiring anaerobically. And if the RQ is less than 1, then the organism is respiring aerobically. In theory, carbohydrates give an RQ of 1, while fats give an RQ of 0.7 and proteins give an RQ of 0.9. Proteins is not often used as an energy store, so an RQ of around 1 usually indicates that a lot of carbohydrates is being used in cellular respiration. RQ of less than 1 indicates that a combination of carbohydrate and lipids are being produced. And it is the end of the topic. I hope it was useful.